We are rolling, so whenever you're ready.
Okay, so I did my thesis on the competitive relationship potential between small mammals and ants, focusing on areas here in the San Juans and the Front Range. So to start, just general, what is competition? Competition is generally the contest between individuals or groups for territory, a niche, which is the location of resources, resources and goods, mates, leadership, etc. Things like that. Two kinds of competition. Intro-specific competition occurs between the same species, where they're competing for resources in an ecosystem. And inter-specific competition occurs between different species, two or more species competing for the same resources in an ecosystem. There are three mechanisms of competition. Interference competition, pictured here, is where two or more organisms compete aggressively for access to shared resources. Exploitation competition is where the use of a shared limiting resource, which is a resource that occurs in small quantities, depletes the amount that's left for others. That's the type of competition we're looking at here. And the third is apparent competition, where you have two or more species that are being preyed on by the same predator. So as prey species 1 increases in abundance, the predator species also shoots up, which leaves more predators to feed on prey species number 2. So kind of interesting one. But yeah, so we're focusing on interspecific competition between ants and rodents, and that's exploitation competition. So other work that led to this project, work done in desert ecosystems by Brown and his colleagues, was focused on competition because seeds are primarily a limiting resource in these environments. There's competition in these environments between small mammals, birds, and ants. However, Brown et al. Looked only at ants and small mammals, as birds are kind of hard to keep out of the exclusion plots that they made. So they took out mammals from plots, ants from other plots, and kept some plots as control and removed both from other plots in order to see how the abundance ratios change in those. And they looked at small mammals and ants because they consume seeds in similar ways. They cache them, consume them, and they disperse them throughout the environments. In tropical ecosystems, you have work done by Danie primarily, is what led to this project. Work done in the Philippines where they had speculation about just general things that affect abundance and species richness patterns across elevational gradients. And one of the potential hypotheses that he posed was this competition between ants and small mammals. He noticed that at lower elevation sites where ants are in high abundance, they were oftentimes the first organism to get to the bait in the small mammal traps and consumed it almost 100% of the time. And then mammals were only abundant in areas where ants were rare or in low abundance, or not present at all in these tropical regions. So that led to this project. So rodents and ants are both resource generalists. They have the capacity to be generalists and fill many species, many niches within an ecological community. They consume plant material, such as leaves and seeds, as well as insects and carrion, which is flesh material. You have some species that are generalists in both of these families. We don't know what species are present in for ants in our sites as of yet, because their identification is still being done. However, the mammals, we know that we have Sauridae family, which are shrews, who are primarily insectivorous, and the Heteromyidae family, which consists of kangaroo rats, kangaroo mice, and pocket mice that are granivores, which feed primarily on seeds. Most of the other species switch to different food resources as they become available in different seasons. And looking at the temperate region, which is where we did this project, the temperate region, there's a greater variability and availability of food resources year-round. And as such, there's less of a need to be specialists, as in desert sites with Brown's work, and so more generalist species of ants and rodents abound. So, is there a resource battle in this? Looked at 3 questions. The first and primary question was looking at if there is a signal of competitive interaction between ants and small mammals. And we predicted that where one group increases in abundance, the other group will decrease in abundance, as shown in this figure, just a model of what you would expect to see, where ants in green and mammals in red where ants are occurring in high abundance values, on the y-axis, you have mammals occurring in lower values. Second question we looked at was, does elevation have an effect? Our prediction in this would be the abundances of both mammals and ants would decrease as elevation increases. And that general model looks like that figure there. And the third question we looked at, kind of briefly, was climatic factors that affect abundance, primarily looking at temperature and precipitation. And we predicted that the abundances of ants will increase as temperature and precipitation increases. And we looked at the elevation and climatic factors to understand other possible drivers of the abundance patterns found in this data that could be separate from the competitive interactions. So what we did, this project was taken out of a larger project made of 4 transects. 3 of them were used in this study. And we had the 2 transects in the Front Range Mountains, they're on the right, and the top transect in the San Juan Mountains was used in this project. We had 8 sites along each transect, chosen every 200 or 300 meters along the transects between the lowest and highest elevations on the mountains. Usually these sites ended up being between 1,500 and 3,700 meters. And we picked sites that were representative of all the habitat types in the area. Those habitat types we categorized in 5 different categories. We had riparian zones, where they have a body of water, a stream or a pond that runs through the site, or evidence of this water in some of the drier sites running through at certain times of the year. Meadow areas, which were open spaces with lots of grass and sparse tree cover. Forests dominated by trees with little underbrush. Rocky areas, which were sparse in flora and exhibited very loose rocky dirt. And then tundra areas, which were open habitats with no trees and located at higher elevations. We had 150 flags along this transect, and each of these habitat types kind of proportioned out to represent each one according to what was actually present. Along each transect, we had vegetation plots spaced throughout. We had about 20 vegetation plots per site, spaced around every 7 flags of that 150 flag transect. And we took various vegetation data at each of these vegetation plots, but the ones that were important for this study were the pitfall traps, denoted by the white circles on the top veg plot diagram, located at east and west at a 3-meter radius from the center of the veg plot. These pitfall traps, we had 2 per veg plot, which led to 40 per site. And then these were cups that were dug into the ground and laid flush with the ground, filled with ethylene glycol, which is low toxicity antifreeze, that green stuff in that picture. And these were flush with the ground with shims used as drift fences to guide insects and other small mammals like shrews into the cups. And for mammal trapping, we used Sherman traps, which are these silver metal boxes, pictured there on the right. And these Sherman traps, we placed 2 per flag. We had 150 flags per transect, so it's 300 traps per site. We had bait on the right side of the transect with seed scented with vanilla, and on the left side of the transect, we baited the traps with a peanut butter and oat mixture to catch these animals. We took measurements in the field, the sex, reproductive status, weight, species, and tag number if it was something that we had already tagged, or we recorded the new tag number if it was a new capture. And if we couldn't determine the species in the field, then we collected the specimens for identification in the lab. Pitfall collection occurred every few weeks for some of the easier to reach sites, or monthly for some of the harder ones. And we collected them in the field with recording of any damage done to the veg plots along the transect, mammal damage, etc. Then we cleaned samples of the ethylene glycol and any debris that was present, so that we could then sort them into these 5 categories. Formicidae, which consisted of ants, orthopterans, which were our grasshoppers and crickets, lepidopterans, which were moths and butterflies, carrion beetles, and then everything else was kind of set aside. And all of these we took counts of everything except the everything else category, stopping at 50 if we had 50 plus individuals. And then we took dry weights of all of these categories, and they were then stored in ethylene in containers with ethanol for long-term storage. And then data analysis. In order to analyze my data, I decided to use abundances of both ants and mammals. For abundance, I use the proportion of pitfall traps containing ants, incorporating all of the traps total over the entire 3 years of trapping. We consider this to be the best method for looking at the densities of ants present at each site. And there was lack of an accurate count, but count was deemed to be inaccurate as individual ants aren't the ones competing with mammals, it's more large densities competing with mammals as opposed to the individuals themselves. And we also didn't use dry weight because of the disparity in sizes of ants. There's a great range in the sizes of ants overall, but in our sites we saw also a great range in the sizes. And there's the ability for a lot of really small ants, which weigh nothing on our scale in the lab, to take in a lot more resources than 2 large ants, which register weight on scale in our lab. So I figured that was not a good thing to use for ant abundance. For mammal abundance, I used the minimum number of mammals known alive, which was the number of mammals that we actually trapped in both the pitfall traps and the Sherman traps. This was used because the minimum number known alive is the mammals we know are definitely there, which could provide a baseline for what is the minimum number of mammals that are contributing to these potential competitive interactions. And then we took average elevation was recorded at each veg plot. So 20 veg plots per site. We had an elevation for each of those. It was averaged out over all of them, and that was taken as our elevation for each site. And then we used bioclim data for the temperature and precipitation data for elevations recorded here in Colorado. And then I used to analyze my data Spearman's rank order correlation, which is a non-parametric test where the data is not required to be in a normal distribution. And I used this because our study had ordinal data as opposed to raw numeric data. So we used the ranking of values to assess the monotonic relationship, the increasing or decreasing of these 2 variables together that did not have to be linear as it would have had to be if I used Pearson's correlation test. So what I found, for ant abundance and elevation, we expected a negative correlation as shown in the top left corner. We found that there's a slight negative correlation over all of the transects but the boulder transect in blue. The boulder transect had a slight positive correlation. None of this data, however, was statistically significant as shown by the p-values there on the bottom. For mammal abundance and elevation, we also expected a negative correlation as well for this, and there was a slight negative correlation of all the transects taken together. But it was also not statistically significant, and the only boulder transect was also the only one to present a positive correlation, which was interesting. And looking at the graph, we had peaks in mammal abundance at 2,200 and 3,000 meters for the Boulder and Big Thompson transects, as well as drops in this abundance for both of those transects at 1,800 and 2,500, and then after 3,200, they start to drop off in the Boulder and Big Thompson transects. What's interesting is the lizardhead transect in the brown triangles there is kind of is opposite in those. It has its peaks where the other 2 have their limits, and which was interesting. And yeah, but overall we did expect to see a negative correlation, and there was a slight one that was not statistically significant. For comparing our ant abundance and mammal abundance, we expected this to be, if there is a signal of competition between these 2 abundances, there should be a negative correlation between the 2. There was no relationship between the 2 at all. All of the Spearman ranking values were effectively 0, with p-values that were very close to 1, indicating no relationship whatsoever between the 2 variables. So that shows we have no signal of competition between the 2. Looking at temperature and precipitation, ant abundance and temperature, we expected to see a positive correlation, and there was a slight positive correlation between the 2. But again, it was just statistically insignificant. And then for precipitation as well, we expected a positive relationship. There was actually a slight negative correlation between the 2 variables, which was also statistically insignificant. The only one that had a positive correlation was the Boulder transect in this set. So, is there a resource battle then between ants and small mammals? Looking back at our questions, is there a signal of competitive interaction between the 2? we predicted that where one group would increase, the other would decrease, and we found that there was no direct evidence for this competition in the study. Our second question, looking at elevation and the effects of elevation on both of their abundances, we found that there was a consistent presence of ants as you moved up in elevation, and a negative correlation for mammals overall, but these were both statistically insignificant and it wasn't that drastic. And then for question 3 are the climatic factors of temperature and precipitation affecting the abundances. There were no statistically significant results in our findings for that. So, looking at questions 2 and 3 initially, the lack of elevation and climate effects in our data. Prior studies along elevational gradients indicate negative correlation between abundance and elevation, and a lot of these studies have also been done for species richness, indicating similar trends as you move higher. Support for these findings has been based on climatic factors, is mostly what these scientists have been looking at, where species richness and abundance have been found to increase in areas with high productivity. Areas with high productivity are areas that have generally high temperatures, high precipitation values, leading to a lot of vegetative material there. And then other things, Kaspari and his work indicates that ants are thermophilic in nature, which means that they thrive at high temperatures. So there should have been that positive correlation between the two. And no significant data in this study supports any of these trends that I found in the research. However, there is also nothing negating these patterns in our data. Then the main question, no signal of competition was found in our data. Potentially why? Was there no signal of this competition? In desert sites, as I've said, they have a limiting resource of seeds. In temperate regions, there's potentially not this limiting resource. Everything is able to go into their own niches, focus on the food that they prefer, and not overlap in competition for these resources. And then in the tropical sites that Danie was at, there were high abundances of ants primarily at lower elevations, and mammals weren't able to expand in abundance until ants were gone completely or rare in some of the higher elevation sites. So the relatively low abundances of both ants and small mammals in these sites lead to less incidence of competition due to the lack of overlap in their ranges potentially. And then very few studies look at this relationship between mammals and ants still, even though a lot of studies that I found in the literature have posed the possible question of there being competitive interactions between the two, but there's still nothing done other than Brown's research in deserts on this relationship. So there's potentially minimal competition in these temperate regions. Temperate regions offer more variety and consistent abundance of food due to the higher productivity in these regions. And they allow mammals and ants to move into the niches that they prefer. So you have deer mice, which are very abundant, they're Paramiscus species, and harvest mice shifting into their preferences for leafy material and insects, leaving seeds open for the generalists, for the specialists, kangaroo mice, kangaroo rats, and pocket mice. Then you also have ants as well, are largely preferred to be a generalist species, where I'll wait for Barbara to come in. Barbara had to step out because there are lots of noise in the hallway. So we're gonna wait for her to stay.
People coming down the stairs, I can't yell at all of them, I have to stand there. I don't know where that is, other people in the world, but
Okay. So, then ants are also generalist species who prefer to feed on nectar, leafy material, scavenging, carrying some fleshy material around, and as well as capturing prey, in addition to feeding on seeds that were the primary resource in desert sites. We also had, some of our sites had damage to some of the pitfall traps due to marmots, bears, other small mammals coming in and destroying some of our veg plots, so that limited some of our sample size at some of our sites. But, areas for future expansion in this. So we ruled out using abundances as of ants and mammals to find a signal of this competitive relationship. But there are other ways that the data could be manipulated, looking at like redoing the project, if I was gonna redo it again, I would potentially do exclusion plots in these temperate regions, similar to what Brown did in his studies, where you remove ants from certain plots, mammals from other plots, and you keep some plots as control and remove both from others, and see how the abundance patterns change in those plots relative to each other. Or you could do the same trapping project in the tropics, where there is higher species diversity, higher abundance patterns seen in a lot of these, as well as that stark contrast that he saw in his study in the Philippines, where you have a lot of ants at lower elevations and then mammals at sites where ants are not present. So seeing that dichotomy could be a way that you could see this. As well as a way to look at it with our study, limiting the study to specialists only, which would be interesting we know the species that we have that are granivores or insectivores primarily, and as soon as the ants are ID'd for species, we can potentially do that as well in our data. And then I thought it'd be interesting, just thinking about it, looking at traps that were located near ant colonies, and seeing potentially the mammal activity that was present there. Outside of this study, competitive interactions between distant relatives and individuals with drastic size differences are rarely looked at in the literature. And this is interesting to me, considering ecological communities are made up of these complex webs of many interactions between every single member of that community potentially. And understanding these interactions could have a lot of conservation implications, as ants and rodents, both global species who are abundant and able to move into a lot of different niches, expand as global warming occurs. They can expand up higher into different ranges that they were originally outside of their historic ranges, pushing out a lot of other organisms. And if we don't know the potential competitive interaction between these keystone key species and other ones, then we won't know them. So yeah, that's my thesis. Cool.
Thank you very much for your summary. And this is the time when we thank you for being here and ask you to leave.
Thanks, guys.
Let me just turn this recording off.